are biocatalysts present in the cell. They help in metabolic reactions. Many metabolic reactions takes place with the help of enzymes. Therefore, enzymes are called as biocatalyst. Catalyst means they promote the enzymatic reactions, and these reactions taking place inside the cell cytoplasm. Anabolic reaction and catabolic reaction takes place inside the cell cytoplasm, and these reactions requires the presence of enzymes. Enzymes are made up of proteins. In the enzymes, which molecules are there? Protein molecule. Okay. First, enzymes are biocatalyst. Enzymes made up from protein. Enzymes catalyze many chemical reactions inside the cell. Nature of enzyme, proteinous, that means enzymes are made up of proteins. Properties of enzymes, there are near about major 6 properties of enzyme. First property, enzymes are proteinous in nature. That means these are made up of which molecule? Protein. Enzymes have 3-dimensional conformation, 3-dimensional structure. Then enzymes have catalytic property. Catalytic property means when enzyme reacts with substrate, they produces product, and again enzyme gets separated. That means enzymes have catalytic property. Then enzymes act on specific substrate. These enzymes act on specific substrate. If there is the presence of specific substrate, then enzymes react. That is called as specificity of action of enzymes. The next property, reversibility of action. What is meant by reversibility? When E plus S is equal to P plus E, again P plus E can produce E plus S. That is called as reversibility. This is also one most important property. Then acts at a specific temperature. Enzymes acts at a specific temperature. That means human body temperature is near about 37 degrees Celsius, and that temperature, human body temperature, is required for human body reactions inside the cell. In the human body cells, enzymatic reactions takes place at maximum speed. At which temperature? 37 degrees Celsius. When temperature goes down, enzymatic reaction slows down. When temperature is increased beyond limit, enzymatic or metabolic reaction gets stopped. Is it clear? That means specific temperature is required for enzymatic action. Optimum temperature at which maximum speed is there. Below the limit, reactions get hampered. Above limit, reactions get hampered. That means enzyme acts at specific temperature. These are some properties of enzyme. Then next, this is the common reaction or equation. E plus S at specific temperature. P means product. E means enzyme. S means substrate. Substrate means the substance on which enzyme act. That is called as substrate. And the product, that means the after reaction the product is produced, that is P. What is the classification of enzyme? First, oxidoreductase type of enzyme. They acts on, example is there, alcohol plus NAD produces aldehyde and NADH2. That means from alcohol H2 is removed, therefore called as reductase. Oxidoreductase, hydrogen is removed and added to NAD, therefore NADH2 is produced, and from alcohol which product is produced? Aldehyde. By the action of which enzyme? Dehydrogenase. Which enzyme? Dehydrogenase. D means removal. Hydrogenase, that means removal of hydrogen. Therefore, from alcohol H2 is removed, added to NAD. Therefore, NAD is converted into NADH2. And from alcohol, which product is produced? Aldehyde. The next type of enzyme that are called as transperases. Transperases, for example, glucose plus ATP converted or transferred into glucose 6-phosphate plus ADP. What is meant by ATP? What tell me the long form of ATP? Adenosine triphosphate. When tri means 3 phosphate, adenosine plus 3 phosphate. From this 3 phosphate, 1 phosphate is removed and there is a adenosine diphosphate is remains. That means di means 2 and tri means 3. ADP is produced and 1 phosphate from ATP is taken outside and attached to glucose. Therefore, glucose is converted into glucose 6 phosphate. 1 phosphate is added to the glucose at 6 carbon atom. This is a glucose ring. In the glucose ring, there are 6 carbon atoms are there, and at 6 carbon atom there is an addition of 1 phosphate. That is glucose 6 phosphate is produced. That glucose 6 phosphate is produced from glucose by taking out the 1 phosphate from ATP and added to the glucose. That is transfer of 1 phosphate to the glucose takes place. Therefore, this enzyme is called as transferase. This enzyme is called as transferase. Why transferase? Transfer 1 phosphate to the glucose. Therefore, glucose is converted into glucose 6 phosphate. This enzyme transfers 1 phosphate group and enzyme glycokinase. That enzyme is transferase enzyme. Example, glycokinase takes part in this reaction. The next type of enzyme, that is hydrolases. Hydro means water. These enzymes act in the medium of water, H2. Sucrose. Sucrose is converted into 2 molecules, that is glucose and fructose. खायची जी साखर असते त्यात पाणी ओतलं की 2 मॉलिक्यूल तयार करतात. खायच्या साखरेमध्ये सुक्रोज म्हणतात त्याला. पाणी ओतल्यानंतर त्याच्यापासून 2 मॉलिक्यूल तयार होतात. कोणते कोणते? सुक्रोज आणि फ्रुक्टोज. Which 2 molecules are produced? Sucrose and glucose and fructose from the sucrose. By which enzyme? Sucrase. Sucrase is which type of enzyme? Hydrolases. Which type of enzyme? Hydrolase. Then next type of enzyme, liases. Liases, example, histidine enzyme acts on histidine and histidine decarboxylase takes T means removal and carboxylase means CO2. From histidine CO2 is removed. That CO2 is removed from histidine and the compound produced from histidine is histamine. Which compound is produced in the body? Histamine by removal of CO2. Which enzyme takes part in this reaction? Yes, histidine decarboxylase. Which enzyme? Histidine decarboxylase. Next type of enzyme, that is isomerases. Isomers म्हणजे प्रतिरूप एकमेकांची. Glucose 6 phosphate is converted into fructose 6 phosphate by the enzyme isomerase. Glucose 6 phosphate. Glucose and fructose, these are 2 isomers of each other. ग्लुकोज पण 6 कार्बन ॲटम असलेली रिंग आहे आणि फ्रुक्टोज पण 6 कार्बन ॲटम असलेली रिंग आहे. फक्त ग्लुकोज जर आरशासमोर उभा राहिला आरशासमोर तर आरशाचं जे प्रतिबिंब आहे ते काय असणार आहे? फ्रुक्टोज. फक्त दिशा चेंज होते त्यांची. लक्षात आलं? जसं आपण मिरर म्हणून ग्लुकोजचं आयसोमर कोण? फ्रुक्टोज. मग आयसोमरेजेस एन्झाइम काय करतात? ग्लुकोज 6 phosphate is converted into fructose 6 phosphate. And last type of that, these are the classification of enzyme. First, oxidoreductases, transferases, hydrolases, liases, isomerases, and last type of enzyme, that is called ligases. Ligase means जोडणारा. 2 पदार्थ जोडणारा एन्झाइमला म्हणायचं लिगेज. काय म्हणायचं? लिगेज. ठीक आहे. Here we complete